Hey guys, so I am going to try to figure out this DNA stuff, and I thought you could join me. So the first thing I did was I asked, what are the best DNA samples for accuracy in forensic analysis? And the obvious things popped up: blood, saliva, buccal swabs, tissue samples, hair follicles. Then I asked, are fingerprints good for DNA? Fingerprints themselves do not contain DNA. Okay, yeah, that was kind of a bit poorly worded question. They are unique patterns formed by ridges and grooves on the skin's surface, but fingerprints can be valuable for identification purposes and also for touch DNA. What is touch DNA? It's when a person touches a surface and they leave behind skin cells or other biological material that may contain DNA, and this can be analyzed. The reason why I started looking at this is because I said something in my last stream, which was, well, STR is likely fairly reliable. And if they extracted DNA from a bone from 30 years, a 30-year-old bone that was outside, it shouldn't be difficult to do it from touch DNA. But that is an incorrect conclusion. And that's why I think it came back up to me, because it's not the same. The length of time and what they're extracting it from, I mean, I'm sure it's difficult to do that, I would imagine, but it's just not the same thing. Because touch DNA, what we're really talking about is, some skin cells, right? Unless you have some like oil on your finger because you've been perspiring, or maybe you cut your finger and there's a little blood or something. But outside of that, most people with clean hands, if they touch something, their touch DNA is left. Really, what it amounts to is some skin cells. So then I asked, how long will touch DNA last before it starts degrading? And I think, we went over this before, but essentially, if the temperature is high, if it's hot, it's going to degrade faster. If it's humid, it's going to degrade faster. If exposed to light, it's going to be a problem. Which theoretically, in that house, it was cold because they left the door open, they said, and it was a cold day. So different surfaces can retain DNA for varying lengths of time. Non-porous surfaces tend to preserve it longer. Now, we know it was left on a brass snap, I believe. How porous are snaps? Touch DNA can remain detectable for days to weeks under ideal conditions, but it may degrade within hours in adverse conditions. Okay. So how likely is it to get a match in a genetic database with touch DNA? While touch DNA can lead to successful matches, it is generally considered less reliable. Therefore, corroborating evidence is often necessary to strengthen the case. Interesting. So touch DNA in and of itself is kind of like what we were talking about yesterday, which was when we were talking about the odds of somebody else having the DNA, your DNA. What I'm starting to realize is, for whatever reason, I'm thinking that a genetic profile and your DNA are the same thing. And from what that article said yesterday, they're not. They're two different things. And when they're getting your DNA, they're really just getting a portion of your DNA, it sounds like. Because when they talk about loci and then they have certain patterns that they look for to make it more or less likely that you're this person, that means that they don't always have all those other things. For example, STR has more markers. They build out more genetic markers. They're using, I guess, what you could call genetic markers. Whereas, I'm sorry, that's STR is not. It's not using those. It's just, you know what? Let me find out. What does STR DNA test for? So STR testing focuses on particular locations, loci, on chromosomes that contain repeat sequences. The number the number of repeats can vary between individuals. So there are specific locations that they look at on your chromosomes under a microscope, obviously. And let's say how many locations there are, we know there's at least 13. They look at those and then they look for the pattern. So I'm guessing, if you have a pattern of A, B, G, D, A, C, or whatever it is, and it repeats a couple of times, right? And then in another loci, you have another one, and then somebody else matches those two, right? That seems like it would be like insane and impossible, but it's obviously not because we share a lot of genetic material with even with even primates. So STR profiles are unique. So what they're looking for is the number of repeats on certain locations. They're unique to each individual, except identical twins, making them useful. Comparison testing allows for the comparison of DNA profiles from different sources. Okay, yeah. And then it depends on the database size. Okay, so how many locations are mapped out? How many locations, loci, are mapped out on how many locations are mapped out on a chromosome? It can vary depending on the context, but in forensic context, 13 to 20 specific loci are frequently analyzed for. For example, the FBI's CODIS database uses 20 loci for its DNA profiles. So 20. The human genome contains thousands of loci, but not all are used in forensic testing. Researchers may map hundreds or thousands of loci across the entire genome for genetic studies. Okay, so here's my next question then. So the max they have, it sounds like, is 20, and FBI CODIS uses 20 loci, but there are literally thousands, right? So what is the difference? Actually, no. So then do SNP DNA tests map more loci? Okay, so that's the main difference between the two. STR, which is what's the standard, and it's for the database that has criminal offenders in it, use 20 loci. Others use 13 to 20. And we were talking yesterday about cases where if they have a 9 loci match, they're calling it their person. This is starting to look really bad, to be honest. But let me keep reading to see. SNP tests. These tests can examine hundreds of thousands to millions of SNPs across the genome. This high density of loci provides a more detailed genetic profile. Okay, that makes sense. These are variations at single nucleotide positions in the DNA sequence and are the most common type of genetic variation in the human genome. So SNPs are capturing genetic variations, the most common ones, though. Okay. Often used in ancestry testing, population genetics, and complex trait analysis due to the large number of loci. All right, so this kind of explains what I was talking about yesterday as well, because when you do genetic testing, it's an SNP clearly, and that's how they're able to gather all this information. And it's also why your ethnicity may change by percentages over time, because the more people of that ethnicity that get into the database, the more representative, the higher your sample size, the more accurate it's going to get. All right, so here's my next question then, because they didn't say anything about SNP testing for forensics. So what are the limitations of using an SNP profile to determine leads in a criminal case? Wow, this is also really bad. SNPs are generally less variable than STRs, making SNP profiles less effective for distinguishing between individuals in forensic contexts. This can lead to a higher chance of false matches. Certain SNPs may be more common in specific populations, which can complicate interpretation and lead to biased conclusions. How are they getting away with using SNP? You know what they need? What they need is a scientist who's not forensic, because they're not going to do it. A scientist who uses these SNP profiles to study, medical things, to come in and say that these SNP profiles are not helpful in determining suspects due to the fact that there's too high of a risk for a false match. This type of DNA was utilized to create genetic lineage lines, and then study for medical conditions, not for forensics. And that's what they're doing. And I think that argument needs to be made. In my opinion, it would be a helpful argument to make. It's probably a bigger, it's easy for me to sit here and say this. It's overwhelming what they are fighting against, because this SNP, they're trying to make this a standard thing that you can utilize to incriminate people for things like murder. And we're reading right now that SNP really isn't helpful in forensics. The use of SNP profiles can raise ethical concerns regarding privacy and potential misuse of genetic information. SNP testing can be more expensive and time-consuming. Okay, how can these limitations be mitigated in practice? Okay. All right, so fair enough. I'm a little more calm now. Using both SNP and STR testing to leverage the strengths of each. STRs provide strong individual discrimination, while SNPs can offer additional genetic information and context. Unlike robust statistical models to interpret SNP, taking into account population structure and allele frequencies, this helps in making more accurate probabilistic assessments. So you definitely need training for forensic analysis of this, clear guidelines, and then public awareness. Okay, how about this? What is the accuracy of a STR, I'm sorry, a SNP profile that was developed using software starting from a partial touch DNA sample that yielded no matches. So the accuracy can be influenced by several factors. The quality of the sample. So we already know touch DNA is likely just a couple, maybe one or two skin cells. The more SNPs included in the analysis, the greater the potential for accurate profiling. However, if only a limited number of SNPs are analyzed, the results may be less reliable. The accuracy of the SNP also depends on the algorithms used by the software. And this is what I was talking about with Ancestry.com. One day you're 40% Italian, the next day you're 28% Italian. That just shows you right there, okay, that what they're saying, that's how the algorithms are developed and how you're funneled. So you're being classified by this SNP because they can't have all of his thousands of millions, right? What if they got another 20,000 on him? Like if it's 5 years from now and everybody's got 20,000 more and you recheck this 5 or 10 years from now and you see that it's not even close to being him. The effectiveness of matching depends on the size and diversity of the reference table. A small or non-representative database may lead to no matches, even if it's accurate. Partial profiles may not provide enough information for a reliable conclusion. Without a complete profile, the chances of finding a match are significantly reduced. And I thought they said it was a partial profile. I have to find where I read how much DNA they use because they did put it out there somewhere. And this sort of works for and against him. It being a partial makes it less accurate, but because it's partial, it's hard to find a match. Therefore, if you have a match, then it's more specific. Population genetics may impact the likelihood of finding a match. In other words, if his ethnicity is underrepresented in the database, and then it'll be less likely to find a match. But that's not it. That's not it here. Hold on. Let me add this to the thing then. What? So my last question was, how can, okay, we asked, what I want to ask it again is here. All right, so how reliable is matching a, okay, how about this? Let's start this over. How do you determine the odds of a DNA match in forensics? I'm wondering if they're matching the SM. So they get this DNA sample, they run it through CODIS. We have no match. They send it to ISP. Maybe that's where they did the CODIS search, whatever. But their STR is not finding any matches. So they take and they give this sample to Othram. Othram runs it through their analysis. I forget the name of it, but it's a software program that instead of you spending hours under a microscope, apparently this thing is able to root out in your DNA. Don't ask me how the fuck that works, because that sounds really complicated and makes me feel like I'm just a bunch of digits. But regardless, Othram does this. And then I'd like to know, question would be how many loci more than CODIS had? Like how many loci were you able to identify? Because we know there can be thousands, millions. So how many loci did they get? And so they did this though. And when they did that, suddenly their John Doe that they put in there started to pop up. Actually, I'm sorry, not with Othram. When they did that, they looked in their databases and their John Doe didn't have any relatives. Okay. So they sent it to the FBI. And when the FBI received it, the FBI looked in databases that Othram has stated themselves in that interview we watched the other day are not okay to do. It's not legal for them to do that. It's really not technically illegal for the FBI to do it, but it's not illegal. It's more or less like it's against their policy. Well, why do they have that policy is my point. Why do they have a policy at the FBI to not look in databases illegally? Well, because some people, like me, for example, who put DNA into a database, opted out of having you look at my genetic information. So FBI gets it. They look in the legal databases, without consent. They look in the databases and they see some relatives. And when they see the relatives, it takes them a little while. It's just like when we're doing detective work online, we see something, we build out a family tree or we build out, groups of people, whatever it is. If you're a cyber sleuth, you know what I'm talking about. So that's what they're doing, but with the help of a computer and a database, right? A limited database. And the name KOHBERGER starts popping up. So then they realize this kid was at WSU. And it sounds like they started looking at him, and then I think they lost him, and then they met back up with him in Pennsylvania. Regardless, they get there, they scout him out. He's like Bruce Lee ninja. I don't know. That's how they're making him seem. And they're taking trash, trying to get the DNA. Finally, they get trash, and they send it in. And the DNA is compared to what? Compared to the original profile, the original STR profile? Or is it just overridden completely and compared to the SNP profile? I'd like to know those two things. I'd like to know which of those two things. And the reason why is because I imagine the accuracy is different depending. And they should have done both. Honestly, they should have checked that DNA from the garbage against the original DNA and against the SNP. And it could result that it makes it more accurate, or it could be that it's less accurate. But either way, we should know that. And then let's say they do both, which is ideally what they do. Well, they did that and they're running it through their genetic database, and a name pops up, Michael KOHBERGER. And then if you have this type of thing, if you're in a database, it'll say like, this person shares this many centimorgans with you. This person is like less than 5% chance of being like your fifth cousin or something. But other times it'll pop up and it'll say, this person shares, 40 or 50% of your genetic material. This person is likely a parent or likely a sibling or likely an aunt. All right. So this pops up and it says parents. So now they know, bingo, this is his father. All right. So we're close. This has got to be him. So that's when they get, right? That's when they get the warrant, they arrest him, and they take a buccal swab. And then the next question I ask is, when they get that DNA back from him, from his buccal swab, which is going to be more accurate than the touch DNA, obviously, do they try to match that with the SDR profile, the SNP profile, or both? And again, that could improve or worsen the accuracy depending on what the results are of all that. But I still think it should be looked at, right? And here's the other thing. And this is why statistics lie, okay? First of all, let me just say this. I believe that DNA does identify many people. I don't think that everyone that has DNA is seen, like, I'm not saying that, oh, DNA is just willy-nilly, like the science sucks or whatever. That's not what I'm saying. I understand that there's a high degree of accuracy with these DNA tests. The issue I have in this case, though, is the many steps it took to make the match. So it's twofold. One is do we want to live in a society where it's okay for them to identify a suspect this way? And the answer to that question for me is absolutely not, right? No. I understand you may be able to find criminals more quickly, and and then this may help, cold cases. Cold cases is a separate issue for me, but even that, I think cold cases I'm just going to leave out of this argument. I'm talking about for an active investigation. Do we want to live in a society where during an active investigation this can be done? The utilization of databases in which people have not consented or are ignorant of the fact that their DNA is being used to help develop leads in a murder investigation. Who's okay with living in a society in which this happens? I'm not. Because people make mistakes, right? People make mistakes. And yes, the science is the science, but we already know, based on the study I read yesterday, no matter what they want to say, that there were more matches than expected. So their percentages are already off. And they're getting sloppier as they're moving along, because now we have this SNP technology in these databases. And even though it was developed, again, intention, right? The intent of those things are for scientists and for families that want to find lost relatives, understand their ethnicity, where they came from better, that kind of stuff. And I think all that stuff is great. But the intention on that versus the intention to, I'm donating my DNA to help law enforcement prosecute criminals, solve cold cases, develop leads in criminal investigations. And hey, they're using it for murder now because that's the yardstick, right? Because if we're not going to use it for murder, we're definitely not going to use it for shoplifting. And we're definitely not going to use it for car theft or anything else, right? So they're going hardcore because they know if we let them get away with this, the other stuff is like whatever. And they will use it for all of those things. They will, because the technology will become cheaper and faster and more accessible and easier. So this is the direction it goes in no matter what, once you say it's okay. And that's where minority report comes in. That's where they find that you're guilty of a crime before you even did it. Reading your thoughts, knowing your moves, knowing your history, knowing your family history. My whole thing is people should be informed about it. So in other words, if you want to donate your personal DNA and consent to medical research, law enforcement leads, solving cold cases, obviously you have every right to do that. And I don't judge on that. I personally would not do it, but you do have the right to do that, right? There are consequences for me not doing it. I may be perceived as selfish. I don't want to help law enforcement. I don't want to help solve cases. I sympathize with bad people, right? Those kind of things. But there's consequences to donating it too, to consenting, because you just never know who or what might pop up at your door for any particular reason in the future, because you're related to a suspect, you have a long lost relative that is in jail, and they there's all kinds of things, right? So like it's a choice, right? But it's a personal choice, not a collective choice. And this is the problem. All of our laws end up getting made as collective choices when luckily in America, most of the time they leave us our privacy, but it's getting like stripped away more and more quickly. And this is just the next step. And so that's my one full problem I have with it, right? Is the accuracy of it and the lack of informed consent. But then the other problem I have with it is just the method. In other words, it'd be one thing if they took the touch DNA off, the STR didn't work, they got the SNP, and then they found their victim or their suspect. There's BRYAN KOHBERGER. Then they go and they arrest BRYAN KOHBERGER. Honestly, like that, I still don't like it. I still don't like it. And I still have the same issues I had before. But to add just another step into this chain where you already are using like a skin cell to get information from. You already did the STR. Now you do the SNP, and then you do the SNP the backdoor way. So like we're really getting ahead of ourselves here, right? Like how many excuses are we going to say? Like, okay, but then this, but okay, but then they checked and they had to do it the back way. But then they did get this hit, but no, it wasn't him. It was his father. So now his father's identified. And so you assume, you've got the right person. And I want to know what the odds are that you have someone who's related to him. What are the odds that he has a brother out there and it's really similar? I mean, I don't know the answers to these things, but I don't think that the chances are 0. And I also don't think the chances are 1 in, what are they saying now? I know I keep making a joke about it, but it's like 1 in octillion. Well, there's only 9 billion people on the planet. So the chances for BRYAN KOHBERGER's DNA to be inaccurate is like double the entire, like almost double the entire planet, or whatever an octillion is, for fuck's sake. That doesn't seem plausible to me, especially because they haven't mapped out the entire genome yet. Like they don't have all of these markers in place. And as much as we are alike, we're still going to have a lot of variations because each and every one of us has one very specific to ourselves. So their math is going to work. That's why statistics lie. Their math will work. But sometimes you have to change the paradigm that you're using and then calculate things. And the paradigm they're using it from is not making sense to me. Especially because all these databases have different numbers of people in them. And this is a select group of people. For example, in CODIS, it's only convicted criminals, right? And then in these genetic databases, look, you could look in 23andMe and compare your DNA to your Ancestry.com DNA, and then compare that to your GEDmatch DNA. And I promise you, you will find that you will have a different percentage of Ukrainian or German or Italian or Korean or Ethiopian or Russian or Irish. You will have a different percentage in each one of those SNP databases, each one. What is the chances that that will happen? 100%. If you don't believe me, look it up. It's 100% that your DNA will give you different ethnicity ranges depending on which database you upload your file to. So what does that tell you? There's no standardization. They're close. It's not like you're going to be 50% something that you only had 1% before. I'm not saying that. They're close, but they're not perfect. And if this science was perfect, if the people reading these charts were perfect, then I would expect it to be, I would expect my DNA to be interpreted identically across all databases. Why are there differences? Why are there such huge differences in genetic databases and people's their traits and their like what's happening there? There's too many confounding variables for them to say 1 in octillion. And I'm not even saying that it's not his DNA. It could be his DNA. I don't know. But they should at least be able to give you a number that's honest, that tells you the chances that based on the fact that this was to begin with touch DNA, which they already said from the beginning, touch DNA needs corroborating evidence. That in and of itself is not enough. So you do the touch DNA, and then they sent it to another lab, and then they sent that to another lab. Then they got his dad. Then they matched him to which one, we don't know. And what if you do all that and you find out, well, the odds are actually 1 in 250,000 or something, or 1 in 2 million. I mean, it's still really high. It's still really unlikely there's anybody else but him. But at least be honest about it. Everybody I've heard say 1 octillion, including newscasters, roll their eyes when they say it because they know it's bullshit. I'm sure there's a calculation that shows up when you look, and there's some really complicated way they do it. But I'd like to see the math proof for that because I don't believe it. Anyway, just wanted to talk about SNP, STR, touch DNA. You know, it's, and the other thing that I heard, I was listening to Pavarotti earlier, and this was like something I missed, where Anne was talking about the location of Brian's car at different times. And this is just unbelievable. Originally, when they got the AT&T warrant back, they had him located at home at his house at 2:44 a.m. based on, I guess, his cell phone data or tower data. I'm not sure. Then when they got the video camera footage back and he was at Bishop Street, allegedly, a white car that they named his, they noticed that it was at 2:42 a.m. And so the very first piece that starts off the whole next hour or so chain of events, actually a couple hours or whatever it is, chain of events with Bryan KOHBERGER, that whole PCA starts off with a fabrication. And that's a fucking problem. Your starting point is already messed up. You got data back and you found out that that car on Bishop couldn't have been his because he hadn't left his house yet. And instead of saying, Maybe we have the wrong guy. You said, nah, fuck it. We'll just change the times. And you changed him home to being at 2:42 a.m. and at Bishop at 2:44 a.m. And that's just a lie, a fabrication. That in and of itself, I believe, is enough to get a Franks hearing. Now, what are you going to do if you get the Franks hearing when you're asked about that? Because that data is still there. Puts him at his home or his apartment with his phone at 2:44 a.m. But now you're saying he was at Bishop at 2:44 a.m. It can't be both. And you chose that second number, 2:44. He can't be at both places at the same time. What's Occam's razor? Well, most likely scenario is the car that you're calling his, that you then follow through on the cameras all the way over to Moscow, couldn't have been his. Could not have been his. Step one, you're already lying. I'm not even going to go through the rest of it because I know people have talked about it, and I'm sure you've caught a lot of this stuff. But that just really pissed me off, and it went right over my head. So I'm glad I listened to Pav kind of bring that up again. It's a really good point. I'm just lost over here in the SNP files, because I don't believe these odds. I've never believed the odds, and I know I'm not the only one. Everybody smirks when they say 1 in octillion. It doesn't make sense, and it's not because it's too complicated. It's because the way it's being explained in court is too complicated, because if they were honest, people would be appalled. Okay, let's just be honest. They would be appalled at the fact that this is what they're doing. And I think it just goes further back to recognizing that the intention for genetic genealogy databases and software that maps out genetic genealogy, the intention was not for forensics. It was for research purposes. And then they commercialized it because they figured out they can make a lot of money, because a lot of people want to know what their ethnicity is. Right? They could have just kept it to themselves and continued researching the human genome, but they needed sample. They needed a sample size. And that's why when you deposit it and you do one of these DNA tests, if they're an ethical company, they ask you, would you like to participate in research? Would you like to help finding missing people? Would you like to help law enforcement? And then you have a choice. But it should be a disclaimer that comes up. I think they need 2 disclaimers, okay? And then I'll stop my rant. 2 disclaimers, but I think all DNA companies should implement. And if they don't have it, then they're not, I wouldn't consider them ethical. You know how, like, before you pay for something, you have to sign that you agree or whatever? It should be a disclaimer. 1, that please be aware that sometimes these results aren't accurate. We may match you with someone that you weren't aware is a family member. This is unlikely and rare, but it can happen. People should know that. And then 2, would you like to opt in for research? Would you like to opt in for LA? Would you like to opt in for cold cases? Would you like more information before you opt in? What does this mean? What does it entail? And then people have a choice. You should have to answer those questions before you even pay for your DNA to be analyzed or whatever. In a perfect world, right? So since it was developed for research purposes and not forensics, how much peer-reviewed stuff is really out there on it? Hey, I could be completely wrong here on my little soapbox. I just started looking into this the past week. Are there peer-reviewed papers out there that show how S&P profiles stand up in terms of accuracy? Have they been studied? They should be able to be studied. if you allow access to the database. I mean, we know that that DOJ representative felt that, prisoners don't, we have to protect prisoners' privacy in the CODA system. So academics don't have any ability to access that research, to research it. But we also know that they don't care about the public out here, us guys. They don't care about our privacy. And trust me, if they needed to use the CODIS to do anything in order to make things less private, it's not about protecting inmates' rights. Like, give me a break. Like, that guy's ever given a fuck about an inmate. Anyway, all right, I'm done. I'm done, guys. Thanks for listening to me rant. I hope you enjoyed it. I will be back later for some more. Let me just put on a song here so we can. Bye, guys.